Hello, my name is Reagan Boyer, and I will be teaching you about cell cycle checkpoints. There are 3 cell cycle checkpoints, and they are intended to prevent cells with damaged DNA from replicating. They are also another way that the body tries to prevent cancer from forming. The first checkpoint is the G1 checkpoint, and that regulates cell size, social signals, and DNA damage. The G1 checkpoint prevents cells that are too small for normal function from continuing on to the rest of the cell cycle. It also allows cells to pass or to remain based off of signaling molecules from surrounding cells, and it also checks for DNA damage. If DNA damage is detected, then the p53 protein will try to repair the damage or trigger apoptosis, which is programmed cell death, if the damage is too great. Any cell that has one or more of these issues will remain in the phase if the checkpoint is functional. The second checkpoint is the G2 checkpoint. Again, it checks for DNA damage, and it also checks to see if the chromosomes have replicated properly during S phase. If you look at the bottom left, the normal cell has correctly duplicated its chromosomes. If you look at the bottom right, the cell that has red chromosomes has incorrectly duplicated its chromosomes. As a result, the G2 checkpoint would be activated, and then the abnormal cell would remain in the G2 phase instead of continuing on. The next phase is during mitosis, and this is the transition from metaphase to anaphase. The metaphase checkpoint regulates and makes sure that the sister chromatids are attached to the spindle fibers, which prevents unequal distribution of chromosomes in the daughter cells. It prevents abnormal cells from entering telophase and cytokinesis. If you look at the bottom left, that shows what a normal transition from metaphase to anaphase looks like. The bottom right shows right here you can see during anaphase that 2 of the chromosomes did not separate properly, and as a result the metaphase checkpoint would be activated, and then that cell would remain inside the anaphase stage of mitosis. Another way that the cell cycle is regulated is the M phase promoting factor, which causes a cell to transition from G2 to M phase, and is controlled by cyclin CDK activity because MPF is composed of a cyclin and a CDK. Cyclins are regulatory proteins, and mitosis is activated when the CDK portion of MPF is phosphorylated, and then mitosis is off when the cyclin is degraded by enzymes at the end of anaphase. Cancer is a result of checkpoints that are either missing or they don't work, so the cells continue to divide despite there being a lack of a social signal, or there's DNA damage. This leads to uncontrolled cell growth, and then the cancer cells infect normal tissue and disrupt their function. Furthermore, they use nutrients needed by regular cells. Cancer cells may form massive tumors that can metastasize and spread to different parts of the body. When that occurs, you can get major tissue damage, and if there are enough tumors, they could kill the organism as a result.